Hello students, today let us learn about nitrate and sulfate reduction and their incorporation into amino acids. First, let us start with the uptake and assimilation of nitrate. The ammonium ions produced during biological nitrogen fixation are oxidized to nitrite and then to nitrate by an aerobic microbial process called nitrification. The specialized bacteria which carry out nitrification are called nitrifying bacteria. Example, Nitrosomonas, Nitrosococcus and Nitrobacter. Nitrification involves a two-step process where ammonium ion is first oxidized to nitrite by bacteria like Nitrosomonas and the nitrite is then oxidized to nitrate by bacteria like Nitrobacter. The nitrate ions thus formed in the soil is the most common naturally available form of nitrogen in the soil available to plants. Plants take up the nitrate from the soil and the nitrate is first reduced to nitrite and then to ammonia before incorporating it to amino acids. Thus, the uptake and assimilation of nitrate by plants involves the uptake of nitrate, reduction of nitrate to ammonia and synthesis of amino acids using ammonia. First, let us see the uptake of nitrate by plants. Plants can uptake nitrate from the soil water through its roots. Since nitrate is a small but negatively charged ion, it cannot enter the root cells freely by diffusion and nitrate uptake is by indirect active transport with the help of nitrate symporters in plant cells. The nitrate symporters use an electrochemical gradient established by proton ATPases in the cell membrane to take up nitrate against both the charge and concentration differences across the cell membrane. Nitrate is also transported into and out of the vacuole where it is stored with the help of proton ATPases operated nitrate symporters located in the vacuole membrane or tonoplast. Now let us see nitrate assimilation. Nitrogen assimilation is the conversion of inorganic nitrogen in the form of nitrate ions taken up by the plants from the soil into organic forms of nitrogen like amino acids. Plants cannot utilize nitrate as such and it is first reduced to ammonia before being incorporated into organic compounds like amino acids. Thus, nitrogen assimilation involves reduction of nitrate in two steps. The first is the reduction of nitrate to nitrite by the enzyme nitrate reductase and second is the reduction of nitrite to ammonia by nitrite reductase. First, let us take up reduction of nitrate. The first step in nitrate reduction is reduction of nitrate to nitrite by the enzyme nitrate reductase and it occurs in the cytosol of both root and shoot cells. The enzyme nitrate reductase is a molybdoflavoprotein containing iron and molybdenum that serves as an electron carrier while the nitrate serves as a terminal electron acceptor. Nitrate reductase requires reductant in the form of coenzyme nicotinamide adenine dinucleotide or NADH in bacterial and plant root cells or nicotinamide adenine dinucleotide phosphate or NADPH in leaf cells and fungal cells depending upon the organism. The enzyme contains FAD or flavin adenine dinucleotide as its prosthetic group which is associated with molybdenum. The enzyme nitrate reductase is a highly regulated enzyme and its synthesis is induced by the presence of nitrate while its activity is stimulated by light. Light, carbohydrate levels and other environmental factors activate nitrate reductase by stimulating a protein phosphatase which dephosphorylates certain residues of nitrate reductase leading to the activation of the enzyme. Recent findings suggest the enzyme nitrate reductase to be a complex enzyme with 8 S unit in which NADPase cytochrome B557 reductase occurs as a separate subunit which is bound to the nitrate reductase by a molybdenum binding protein. The transfer of electrons from reduced coenzymes to nitrate involves two steps where electrons are first transferred from NADH or NADPH to cytochrome B557 through FAD catalyzed by NADPH cytochrome B557 reductase, and the second step involves the transfer of electrons from cytochrome B557 to nitrate through molybdenum catalyzed by nitrate reductase. Since cytochrome B and molybdenum are 1 electron carriers, 2 molecules each of cytochrome B and molybdenum are required to transfer 2 electrons through them, releasing 2 protons when electrons are transferred from reduced FADH2 to cytochrome B, and these protons are the last step of reduction which results in the formation of nitrite and water. Next, let us see the reduction of nitrite to ammonia. It is the second step of nitrate reduction involving the reduction of nitrite ions to ammonia, and it occurs inside chloroplasts in leaf cells and leucoplasts of other cells. The reaction is catalyzed by the enzyme nitrite reductase, which is a metalloflavoprotein that contains 2 prostatic groups, which are an iron-sulfur cluster and a specialized heme. The immediate source of electrons for the reduction of nitrite is reduced ferredoxin in chloroplasts. It also requires reducing power in the form of NADH in bacterial, fungal and plant root cells or NADPH in photosynthetic cells. The transcription of messenger RNA for nitrite reductase enzyme is induced by nitrate and light, which is stimulated by sucrose, while asparagine and glutamine repress it. In unilluminated cells, FAD serves as an electron donor instead. The reduction of nitrite ions by nitrite reductase results in the formation of ammonia as the product. Let us now see the synthesis of amino acids using ammonia. The ammonia thus formed is highly toxic and are used for the synthesis of amino acids, which represent the initial products of nitrogen assimilation. Amino acid synthesis from the ammonium ions commonly take place near its site of generation, and it occurs by the methods of reductive amination, amide synthesis and other secondary pathways like transamination and carboxyl phosphate reactions, and is similar with the ammonia assimilation in nitrogen fixing microbes discussed in the previous lecture dealing with biological nitrogen fixation and ammonia assimilation. These reactions result in the formation of glutamate, glutamine and carboxyl phosphate. The nitrogen of carboxyl phosphate are used either in the synthesis of pyrimidines or the synthesis of arginine, whereas all the other amino acids are derived directly or indirectly from glutamate or the amide group of glutamine. Next, let us learn about sulphate assimilation in plants. Unlike animals that require essential sulfur-containing amino acids in their diet, plants are able to assimilate inorganic sulfate ions to fulfill their nutritional sulfur requirement. Higher plants require sulfate ions for the synthesis of different metabolites and coenzymes. Sulfate is taken up actively across the plasma membrane of the root cells and is transported to the shoots through the xylem vessels. The uptake and transport of sulfate is driven by a proton gradient generated by ATPases and occurs through a proton or sulfate cotransporter. The transported sulfate is unloaded in the chloroplast of shoot, where it is reduced or they are stored in the vacuole. The transport across the tonoplast membrane of vacuole is mediated by an unknown mechanism that may involve electrical gradient between the vacuole shape and cytoplasm, while the phosphate or triose phosphate translocator of the inner chloroplast membrane, or a proton or sulfate symporter may mediate sulfate transport into the chloroplast. The sulfate taken up by the plants from the soil is either stored in the vacuoles or is incorporated into organic compounds by a process known as sulphate assimilation. Sulphate assimilation commonly involves reduction of sulphate to sulphide, which can be assimilated by plants. Now let us see reduction of sulphate in plants. Sulphate reduction mostly takes place in the chloroplast of leaf cells. Sulphate is reduced to sulphide using 8 electrons as shown in the reaction. The reduction of sulphate to sulphide occurs in 3 steps. First, sulphate is activated to adenosine 5-phosphosulfate or APS catalyzed by ATP sulfurylase. Secondly, APS is reduced to sulphide catalyzed by the enzyme APS reductase using glutathione as the reductant in most cases. Finally, sulphide is reduced to sulphide by sulphide reductase using ferredoxin as a reductant. Next is sulphate assimilation. The assimilation of sulphate starts with the reaction of the reduced form of sulphide with O-acetylserine or OAS catalyzed by OAS thiolase to form cysteine. OAS is commonly formed in the cells by the acetylation of serine with acetyl coenzyme A catalyzed by the enzyme serine acetyltransferase. The formation of cysteine during sulphate assimilation serves as the direct coupling step between sulfur metabolism and nitrogen assimilation in plants. Cysteine then acts as the sulfur donor for the synthesis of methionine through the transsulfuration pathway and the methylation of homocysteine. Methionine is another major sulfur-containing amino acid present in plants, and together with cysteine, they play a role in maintaining the structure, conformation and function of proteins and enzymes, and they are also present in seed storage proteins. Cysteine is further incorporated into proteins and glutathione and serves as the substrate for various other reduced sulfur compounds. The various other sulfur compounds in plants are as shown here. The first one is glutathione. Glutathione or its homologues, example homoglutathione in Fabaceae, hydroxymethylglutathione in Poaceae, are water-soluble non-protein thiol compounds present in plant tissues, and they are synthesized using cysteine as the direct precursor. The synthesis of glutathione and its homologues involves 2 steps of ATP-dependent reactions. First, cysteine and glutamate react to form gamma-glutamyl cysteine catalyzed by gamma-glutamyl cysteine synthetase. Next, gamma-glutamyl cysteine reacts with glycine or beta-alanine or serine in case of glutathione homologues to synthesize glutathione catalyzed by glutathione synthetase. Glutathione is commonly maintained in the reduced form as reduced glutathione represented by GSH by an NADPH-dependent glutathione reductase, and the ratio of reduced glutathione or GSH to oxidized glutathione or GSSG in plants generally have a value higher than 7. Glutathione plays various roles in plant functioning and works as an important reductant. The next group are the sulpholipids. Sulpholipids are sulfur-containing lipids present in plants. Example, sulfoquinovosyl diacylglycerols present in plastid membranes. The next are the secondary sulfur compounds. Plants also contain various other sulfur-containing secondary compounds like glucosinolates found in Brassica species responsible for the flavor properties of Brassicaceae, gamma-glutamyl peptides and alanines or S-alkenyl cysteine sulfoxides found in allium species, serving as flavor precursors for the odor and taste of allium species, etc. Let us now see the conclusion. Thus, the nitrate ions formed by the nitrifying bacteria in the soil serves as the most common naturally available form of nitrogen available to the plants, and plants take up these nitrate ions from the soil and reduce it to ammonia and incorporate it into amino acids during nitrate assimilation. Plants also take up sulphate as the utilizable form of sulfur from the soil, and the sulphate ions are also reduced to sulfite, which is used to synthesize the amino acid cysteine during sulfur assimilation. This formation of cysteine during sulphate assimilation serves as the direct coupling step between sulfur metabolism and nitrogen assimilation in plants. Thank you.